Now I will turn to the next presentation on the agenda, and I am pleased to present an overview of the research priorities and all of the programs underway today for homocystinuria, which I am very pleased, based upon my earlier comments, has expanded and progressed considerably in the past 5 years. And I think Jan deserves credit for that. Because showing one promising approach to a disease often leads other researchers and companies to get involved because they see that it can be done. I'll also give you an update of the global grants program that I mentioned that we established for homocystinuria just a few years ago. And you'll also hear an update during this hour time frame from both Orphan Technologies and Aglia, as they have two of the potential therapies furthest along in development. So I do want to make disclosures. It's always important for the audience to understand any involvement that any presenter may have with the work they are presenting. First, you're aware that I'm president of HCU Network America, but also after sponsoring the mouse study 40T58 and meeting the company Orphan Technologies that's bringing it forward, I was asked to join their board so that I could help contribute expertise from my many years in the pharmaceutical industry as well as my knowledge of homocystinuria. And I agreed to do that, but to maintain my independence, I donate my board fees, so I don't have any financial conflict of interest to HCU Network America. And I also continue to fund other research projects for HCU. My goal is to have as many treatments available on the market so that physicians and patients have choices and can figure out what is best for any individual patient. Also, I oversee the Hempling Foundation for Homocystinuria Research, which I've already mentioned, and that fund has supported some of the research projects in this presentation. So you've seen this pathway many times. I won't take you through it. I just wanted to set it up as a foundation because I'm then going to come back and point out to you where the various approaches in development fit into this cascade. And you all know this bottleneck here, was that what caused the dam that Kim talked about yesterday, is a major focus. So there are essentially 5 different approaches being studied for HCU. There were a few others that we may have talked about in the past, but these are the 5 where there are active programs underway. So I'll explain these 5, and then I'll go into each one and tell you what specific programs are going on and what is their status. The first is enzyme replacement therapy, which you are aware of. Can you directly replace the enzyme by administering an already produced enzyme to a patient? Second, I don't know why these numbers always mess up when I send you these slides today, but it should say number 2, gene therapy. So instead of directly administering the enzyme, can you instead administer the DNA so that the cells can produce the enzyme? You just create a little manufacturing facility right within the HCU patient. An alternative approach is a bioengineered red blood cell that has the DNA inside. Then there are alternative enzymes other than CBS that can degrade methionine or homocysteine. You'll hear about one of those from Aglia. The next approach, can you activate the enzyme? Many HCU patients still have enzyme available, but because of a defect, they just don't have enough and it's not active enough. For those patients, can you administer a compound that can restore the enzyme function by either stabilizing the enzyme or by activating the enzyme? And lastly, metabolic pathway modification. Can you use nutrition or administer compounds that can address some of the metabolic imbalances that are caused by HCU? So let's go back to the biochemical pathway. And here's where each of those approaches fit in. The alternative enzyme can come into play at the level of methionine or homocysteine. The replacement of CBS, enzyme replacement therapy, gene therapy, or cellular, which is the red blood cell therapy, or activating or stabilizing the enzyme. And then the metabolic pathway can come into play in the top half of this pathway or the bottom part. You know, you heard yesterday that some of the downstream compounds, because of the dam due to the blockage at the CBS enzyme, may be deficient. Can you do something to address those deficiencies? So I have 2 slides now which take you through the programs that fit under these 5 approaches. And first, with the gene and cellular therapy, the first program shown on top, AAV, is the name of what's called a vector. It's similar to the cold virus. Adeno-associated virus is the full name of it, but it has no biologic activity. It's a delivery mechanism, because you can't just inject DNA. I mean, you could, but as Kim knows, you'd have to do something like electroporation, which would be a nice shock to the muscle, and you don't want to do that. So AAV is a carrier vehicle that's been proven. Different forms of AAV have been proven to have uptake in different tissues. So Warren Kruger at Fox Chase, collaborating with Ron Crystal at Weill Cornell in New York City — Fox Chase is outside of Philadelphia — they have collaborated on this. Warren has a mouse model for homocystinuria and is a yeast geneticist. Ron has helped develop many different gene therapy programs for rare genetic diseases. And so through the foundation I directly funded both of their work to do a mouse study. And we do have positive results. You know this. It's not been publicly presented yet. It will be. That's coming soon. But I can tell you the results are positive, and they are now determining the best path forward, which will likely be a biotech or pharma partner that can help then bring this into development. And so when you look at the usual timelines, and this is my estimate based upon the usual industry timelines. None of this comes from the researcher or the company. But based upon where they are now, I think the earliest they could get this into human testing would be 2022. And assuming everything went really well, there you go, Kim, it's more than 5 years away. 2026. Kim keeps saying every time I see her, gene therapy is 5 years away. But we've been saying that for, 30 years. 35 years. But it's getting a lot closer, and there are now products on the market for genetic forms of blindness, and there are products in the last stage of human testing for more systemic diseases, which will be the real proof of concept. And so what that could mean is it could be a one-time injection, and if your cells continue to express the DNA, then that could be a one-time approach. Probably more likely 10, 15 years down the road, and this would be monitored, that you're not producing enough enzyme, you may need to re-administer it. But it is a one-time as opposed to once a day or several times a week. The next program is Rubius Therapeutics. As Danae mentioned, they're a sponsor of the meeting, but weren't able to make it. So they take red blood cells and insert the DNA into the cells, and that is then injected. And the theory there is that the red blood cells will uptake the homocysteine that's in the bloodstream, and the DNA has been expressed into an enzyme. So you've got the CBS enzyme inside the cell, and that enzyme will metabolize the homocysteine and will excrete back into the bloodstream the byproducts. It's really high-tech science. What gives me hope on this is that they are in human testing for PKU. And I'm finding this to be common, that companies will get involved and they'll say, okay, let's go into PKU first. It's a bigger patient population, but let's start at the same time working on this for HCU. So we're really able to more or less piggyback on that. And so as that moves forward, it will give us more and more hope that this would work for HCU as well. So since they're not yet going into phase 1, we don't want to try to estimate timelines, because they probably want to see data from PKU first. Next shown on the chart is OT58. You heard about that yesterday, and Marcia is going to come up soon and give you an update. So all I will do is say that phase 1 patient enrollment began early this year, and based upon usual industry timelines, I have here the earliest market launch could be 2023, but if there's one thing I've learned in 25 plus years in the pharmaceutical industry is that timelines are always wrong, but it's the best guess. Then the approach for alternative enzymes, I'm only showing one of them here, the second one is on hold. The homocysteinase enzyme which I will let Aglia explain what that is. They'll be presenting after OT, is currently awaiting regulatory feedback. Marcia is going to show you a slide on the drug development process, but it basically starts with discovery, or you invent something. Then you have to do a lot of animal studies, which is your preclinical. Clinical means it's in the clinic, patients are getting it. Preclinical, it's in animals. You're figuring out how it works. You're figuring out, is it toxic? You're figuring out, some dose response that you could then extrapolate to humans. And then you have to collect a lot of data, including on your manufacturing process, which for many of these products that are what are considered biologics made from a living cell. This one is re-engineered or something, so maybe it's not considered a biologic, but you always have to submit to the regulatory agencies all of your data on your manufacturing process before they will allow a product to be used in humans. And that's called the IND, or Investigational New Drug Filing. And so Aglia is waiting for regulatory feedback, and then they hope to begin those studies that would enable their IND in first quarter 2020, and I think they'll give you even more of an update soon. So based upon usual industry timelines, the earliest launch for that could be 2025. So when you look at this slide, we're in 2019. 7 years from now, we could have three very different approaches available to physicians and patients with HCU, which I think would be a great advancement for the field. And then the last two categories, the CBS activation or stabilization, Tomas, who I just read comments from, who Jan mentored at University of Colorado, has a program where he's trying to address what's called the SAM binding domain, and I'll show you on the next slide exactly where that fits in, that would activate or stabilize the enzyme. That is at discovery stage, where he's still trying to screen compounds and figure out if any of them work, and if so, which ones work best. And he has both a grant from the American Heart Association as well as from Orphan Technologies. So we don't have any estimated timeline until he gets to the end of that phase of research. And then the last category, the metabolic pathway modification. One of the grant we last gave, or from the global grants process, was to Ken McGlynn at University of Colorado. And he is assessing different metabolic compounds in combination with betaine to see if they will improve the effectiveness of betaine. And he is doing that with a grant both from HCU Network America and Australia, as well as some funding from the Will Hummel Foundation. Some of you may have met Tara Hummel and her son Will, who were at the Boston meeting. A family in Pennsylvania with HCU has a foundation that also supports research. And Ken has shared with me, he presented some data at a recent conference, that he has learned a lot already in his research. He has learned that some compounds do not help with the effectiveness of betaine, such as glycine, he told me, has no effect on that. And he also said that he has learned that formate has a very positive effect and makes betaine much more successful. And so he is in active discussions with different potential partners, as he would like to develop a sustained release formulation to be able to offer formate, which I believe would be an oral product, to the HCU community. He said he also learned that tryptophan blocks betaine effects. So I don't know what tryptophan is in, Kim. Is there anything they should be avoiding to avoid tryptophan?
Well, tryptophan is an amino acid, so
right.
there's certain foods that are more prominent.
Okay.
Turkey, but you shouldn't be eating it, so.
Okay, well, be careful of turkey. It has tryptophan in it, which could block the effectiveness of betaine. So, what's that?
We are safe. We're safe.
You're safe? Okay. That's what Kim said. You shouldn't be eating turkey anyway. Yeah, okay. So, those are the 5 different categories of programs underway. And now I'll go back to the metabolic pathway and have the specific products shown here. So at the level of the CBS enzyme, OT58, the AAV gene therapy program, the red blood cell cellular therapy program, SAM binding domain would act within this part of the cascade. At the level of methionine, there was a program, arimethionase, but that is on hold from a French company. Then homocysteine is what Aglia will talk about. And then the metabolic compounds can act at the top or the bottom of the pathway, as well as some of you are familiar with the taurine study that was done. It was a human trial done at the University of Colorado and a few other places, which affects the bottom part of the pathway. And University of Colorado tells me they still think that a study could be done down the road to show that for patients with endothelial dysfunction, it has a positive effect. But they're not hopeful that that will easily be funded, as that would be a very small group. So I'm also showing this slide. When we started out with establishing the grants process, we developed a research map which helped us to categorize these programs. But we also had a panel of about 10 experts, one of whom was Dr. Chapman, global experts, who we asked for input on all these different approaches in development. And they gave us their guidance on, based upon everything they saw and knew, what do they think the key priorities should be for CBS deficiency? Well, first of all, optimize current therapy. Let's make the best use of what we already have. Can we have better outcome studies? Can we have better quality and taste so therapy is easier to take? Can we know more about how to use supplements? Can we understand more on how to improve quality of life? The middle category, improve CBS detection, which newborn screening is a major focus for HCU Network America, but also over time, how can we detect patients if they're missed by newborn screening, by healthcare professionals across the board being more aware of what signs and symptoms should lead to consideration of HCU. Then in the new treatment approach, for any disease area, if you can have a safe oral therapy that's easy to take and doesn't cause issues in patients, that's always the best approach, and with the hope here that we can avoid or severely lessen dietary restrictions. The advisory group was excited about the potential to replace the enzyme, since that's going directly to the cause of what's causing that dam, either with CBS enzyme or other enzymes that can address methionine or homocysteine. And lastly, the potential for approaches like gene therapy to give you a potential curative approach. So just to finish on the global grants program. So after we established the research map and got input on priorities, then in partnership with HCU Network Australia, we convened a global scientific advisory board, and Victor Kosick leads that. Kim is a member of that, as was Jan Kraus. And we established key priorities with input from the committee. We decided to go out with a call for proposals on new treatment approaches, not try to be specific because there could be approaches we haven't thought of, and then an improved approach to newborn screening. We gave the first grant in September of 2018 that I've already mentioned to Ken McGlynn, and Ken is also using some funding from the Hummel Foundation. We had one proposal for newborn screening, but it had a lot of technical issues, and it was recommended that the researcher retool the approach and come back into the next round of call. We then initiated a second call in April of this year. We got proposals, where we asked for full proposals from 3 projects. 2 are for improved treatment approaches. 1 is for newborn screening. We ended up then getting those 2 full proposals for improved treatment. They were currently under review by expert reviewers and the Scientific Advisory Board. The newborn screening proposal did not come back because the researcher does not think they could make it a first-tier program. They couldn't overcome the technical issues, and we already have 2-tier screening approaches. So hopefully we'll be funding one of these approaches. Our deadline to inform the researchers is the end of December. So additional steps planned with our grants program. We certainly want to generate more funding. To be able to do 1 grant every 18 months is a nice start. But when you look, for example, at what the PKU National Alliance is doing, they're spending, Danae, is it $2 to $3 million a year on research grants. And they're doing that through fundraising in the community. And so we hope over time through our fundraising committee that we're able to engage the community to help us set up larger fundraisers at a local level that could help us contribute more to this grants program. It also helps the up-and-coming researchers get more involved in the field, because if they're able, if their mentor is able to get grant funding to fund their work, they get trained doing this work, and as Kim can tell you, that will have a big impact on what they then do in their future lab. We certainly intend to continue to interact with industry and academia. We make ourselves available to any company that's interested in working in the field to give them advice and input, to engage patients when needed to give them input, or to let patients know that there's a trial that they can enroll in. And lastly, we do hope to expand to other causes of HCU, including cobalamin, MTHFR, et cetera. But with the very small funding base we started with, we had to focus, but we hope as we are able to raise more funds, we can do more. So with that, I'll introduce the other two speakers, but first I'd like to thank my co-sponsor, Tara Morrison. I'm sure many of you have run across Tara on Facebook. She posts often on our Facebook site. If you've never looked at HCU Network Australia's website, you can find a lot of very useful information there. And Tara's been a real strong supportive partner in this effort. Victor is our chair, and Warren Kruger is our co-chair, and Kim and others that are listed here are members of our scientific advisory board. And I also thank the researchers, the universities, the academic physicians, the companies, the scientists, the clinicians, but very importantly, the patients that participate in this research. It wouldn't happen without your support and involvement. And lastly, thank my sisters for inspiration. I hope I've been able to do something to really honor their legacy and honor their lives by helping to stimulate more research so that families don't have to go through the suffering and loss like mine did. So with that, I will first introduce Marcia from Orphan Technologies. We'll need one moment to transfer microphones, and she will give you an update on OT58.